Thank you everybody for logging on this evening. In tonight's webinar, it's entitled Liniments, Balms, and Salves, Advanced Care for Difficult Wounds. We've put together two phenomenal speakers tonight, Drs. Jeff Gertner from Stanford and Alex Wong from USC, who will each be giving expert talks on their research. Now, unfortunately, Dr. Gail Gordillo will not be able to join us tonight as the moderator. She informed us earlier today of a conflict that arose, so I will be handling the responsibilities tonight of introducing the speakers and running the discussion. Each of the speakers will be giving a 20 to 25 minute presentation, which will then be followed by 5 to 10 minutes of open discussion. During the talks, if you'd like to type your questions into the chat box, I'll be monitoring it along the way. And then during the discussion period, I can help facilitate raising the question of the speaker. Or you can save your question and just raise your hand later during the discussion period to ask yourself. Once again, this entire series has been made possible by generous support from 3M. In addition, 3M has also sponsored an interactive case review scheduled for November 17th on how to manage complex wounds in plastic surgery. So please check this out. Okay, so we'll now move on to the speakers for tonight. Our first expert speaker who will be presenting is Jeff Gertner. I think most of us know Jeff, but for a little background, he is on faculty here at Stanford and is the Johnson & Johnson Professor of Surgery, Bioengineering, and Material Science. He is also the director of Stanford's Advanced Wound Care Center and has authored over 300 peer-reviewed publications. He runs an NIH and DoD-funded research laboratory examining how physical stimuli alters human response to injury, and his findings have led to over 30 issued patents. Dr. Gertner has founded several venture-backed startup companies, including Neodyne and Arresto, and he's the founding partner at Tautona Group, an early-stage life science fund that's created novel biomedical technologies that have now been brought to the market. So tonight, he will be speaking on some of his work that he's been doing with non-healing wounds. Jeff?
Thank you, Derek. I assume everyone can see my screen?
Yep.
Okay, so I'm going to speak about some of our research looking for new options for non-healing wounds. Oops, let me just, there we go. So I have, as Derek alluded to, I'm involved with several commercial entities, but none of that will really be mentioned in this talk, but these are my disclosures. So I've been interested in wound healing since I started, not plastic surgery, but general surgery. Primarily because one of the first patients I took care of as an intern was somebody who had a big wound dehiscence. And this isn't that patient. This is unfortunately one of my own wound dehiscences. But you know, the sequence that occurs is kind of amazing, especially the first time you see it, where you have this kind of disaster unfolding. You now have this big, gigantic hole in the middle of, in this case, someone's abdomen. And then with just dressing changes and the tincture of time, the wound slowly fills in with granulation tissue, starts to contract, and eventually ends up, you know, like this little scar here. Looks worse than, you know, the primary closure would have looked, but not as terrible as that big giant hole in the middle of the abdomen that we see here. So I've just been intrigued by how wound healing occurs. You know, it's very different than what we see in the physical world, where, you know, if you get a dent in your car, the dent stays there forever until you sell the car. With wound healing, there actually is an active process where new tissue is deposited, new blood vessels come in, a whole slew of inflammatory, innate, and adaptive immune cells come in, and then they all need to go away, and you end up with that scar. And, you know, I think the more I have been involved in wound healing over the past 15 years, the more I realized that we've barely, you know, scraped the tip of the iceberg of the complexity of it. It's probably the most complex thing that occurs following development when you look at biological processes. And it doesn't always go well. And so, you know, I think we can all agree that lots of what plastic surgeons do are see the consequences of abnormal healing. On the top, you have kind of overhealing, which I'm not going to describe, but I think will be the next topic in this webinar series, where you get scar tissue that is disfiguring, but also dysfunctional. And in the bottom, you have a non-healing wound. And if you add all the different things that occur from both overhealing and underhealing wounds, it's, you know, a multiple billion-dollar healthcare problem. There are large companies, including 3M and Acelity, who are multi-billion-dollar purveyors of products in this area. So it's a very interesting area from both a scientific, clinical perspective, but also from a technology perspective. And as I alluded to, a human wound healing really occurs on a spectrum. You know, normal wound healing, unlike the first slide I showed, usually ends up in things we do in surgery with a linear incision or a linear scar, which is not perfect, but is generally acceptable. On the left, you see a non-healing wound. This is a patient with, you know, very severe diabetic foot ulcer that's about to get a free flap. And on the right-hand side, you see a patient with overhealing, which has, you know, a patient with a burn who's got all the sequelae of burn contractures. So, you know, unlike humans, there are organisms in nature that heal actually not even with scar, like that middle panel, which although is not perfect, is better than nothing. But there are animals that heal with no scar, and zebrafish and salamanders and other model organisms can actually regenerate tissue, and you don't have that sequelae of the patch of scar tissue that is between two normal anatomical regions. And it would be great, and I think, you know, many of us that are in wound healing are interested in the ability of tissues to regenerate, because we could get away from the problems of both underhealing and overhealing if we could truly regenerate. But there's a lot of reasons to think, why would we think that we could do the same things that salamanders and zebrafish can do? And the reason is because all of us have had the ability to scar or to heal without scar in the first 2 trimesters of our intrauterine life. And Mike Longacre and Mike Harrison up at UCSF, when they were working in the early days of fetal surgery, really identified that human fetuses heal without scarring, but they lose that ability when they leave the womb. So a lot of the work that and I'm going to only touch on a part of the work that we're working on, but you know, a lot of things that our lab and others have been investigating is trying to come up with ways to get true regeneration, mitigate scarring, and improve healing. You know, wound healing is actually, you know, a very good area to test out new technologies. There's a lot of wounds out there, so it's very easy to do clinical trials, and that's been my focus really over the past 5 or 10 years, has been trying to get things from the laboratory into the clinic. So I'm going to just, for this talk, I'm just going to talk about underhealing chronic wounds, and I'm going to really even bore down closer to diabetic wounds. That's the 800-pound gorilla of wound healing, is diabetes and diabetic foot ulcers, and leads to amputations and lots of downstream consequences that lead to all those healthcare costs. And when I was at NYU as a junior faculty member, Frank Spencer, who's the chair of surgery, said a patient with diabetes heals about as well as a hole in your pants. And I think that's actually been borne out no matter what the setting. If you're operating on a patient with diabetes, or if you see a patient with a non-healing wound in the outpatient clinic, you just know you're going to have problems with diabetic patients. And so when I started my lab, going on, you know, almost 20 years ago, I really wanted to investigate why was that? Why did diabetes and diabetics heal so poorly? And again, as a surgeon, I think our lens is a little different than basic scientists. I wanted to figure out, can you reverse that? And the thing that I've always been convinced is probably the best way to intervene, short of devices, are small molecules. And small molecules are just chemicals. Most of all the drugs, not 99% of the drugs that are commercially available are small molecules that, you know, that's defined as less than 900 Daltons in size. That size lets things penetrate cell membranes, bind to proteins, and exert a biologic effect. And I think small molecules are attractive because unlike very complicated cell-based therapies or peptide-based therapies, they're being chemicals, they're very resistant to proteases and extreme conditions that we find in the wound. Lots of companies, and this is just, you know, a potpourri of pharma companies. There's a well-established track record of how to make small molecules, and they've worked therapeutically. Unfortunately, there aren't any in the entire wound healing space or scar market. There's not a single small molecule either to improve wound healing or to reduce scarring. So we really started a long time ago trying to think, if we understood the pathophysiology of diabetic foot ulcers, we might come up with druggable targets that then we could use to develop new therapeutics for this space. And so, you know, again, to go back to that first slide, you have to before you can identify a target, you have to understand what's going on in normal and abnormal wound healing, particularly diabetic wound healing. And, you know, again, going back to Frank Spencer, you know, diabetics heal about as well as a hole in your pants. When you ask people, why is that? Oh, it's small vessel disease and this, that, and the other thing. There's a lot of hand wavy things. And we started off our laboratory very descriptively trying to just quantify what's different between diabetic wounds and non-diabetic wounds. Initially in humans, we started most of our work on human wound specimens and then moved into animals, and now we're back in humans. So we had to develop, once we kind of identified that there were defects in this proliferative phase of wound healing that appeared to be related to new blood vessel formation, we then went back to the clinic or back to the lab and said, now can we really tease this out to figure out exactly why do diabetics not form angiogenic new blood vessels very well? And so we developed new models. This is kind of a McFarland flap. We didn't develop it, but we used it to kind of look at regions of ischemia and how they promoted new blood vessel formation. And what you find is you find more new blood vessels in the more ischemic regions. Again, what happens in diabetes, these are littermate mice, and this data is in JVC 10 years ago. These are twins, syngeneic mice. One we've made diabetic with STZ, and one is a wild type. And if you raise that McFarland flap, you see that the survival of that tissue is quite different. You know, you can use lots of different metrics to quantify that, but you know, really the take-home message is obvious that there's a difference between diabetic and non-diabetic in how they respond to ischemia. We also found that this also occurred in aging. So there's some commonality between what goes on in diabetic wound healing and aged wound healing, both related to the ischemia response and neovascularization and the potency of neovascularization in response to ischemia. And so, again, I'm not going to present the detailed data, but our conclusion, you know, about a decade ago was that diabetes and aging impair the ischemia response. And when you're talking about the ischemia response, you're talking about HIF-1 alpha, HIF-1 beta, hypoxia-inducible factors. The Nobel Prize was awarded for this 2 years ago to Greg Semenza and some of his colleagues for really defining how cells know how much oxygen they're in. And it's essentially through something that's not transcriptionally mediated, it's translationally, or it's regulated post-translationally. HIF-1 alpha is constitutively transcribed, and in high oxygen environments, it's targeted for degradation by the proteasomes. In low oxygen environments, those abilities for the HIF-1 alpha to be degraded by the proteasome is limited, and so you actually get increases in HIF-1 alpha that then go to the nucleus and turn on the entire pattern of ischemia response. And ischemia response is not just important for neovascularization in the context of wound healing. It's in all tissues, including tumors, including hearts and brains, but it's a ubiquitous common pathway by which cells sense the oxygen environment to either turn on or turn off the genes that are involved in the ischemia response. So again, I won't present all the data, but this is about 4 or 5 years worth of work where we were trying to understand what is it about diabetes, and in particular glucose, that blocks this hypoxia response. And long story short, what we found was that there was actual glucose poisoning through a molecule called methylglyoxal, you actually got methyl glyoxylation of an arginine residue in this transcriptional cofactor P300 that prevented HIF-1 alpha binding to it, that then didn't allow HIF-1 alpha to bind to DNA and turn on RNA transcription. So you block the ability of these two to bind. You don't get transcription of VEGF, SDF1, and other hypoxia response genes. And so we, once we knew that, we thought, okay, can we reverse this? It seems like that should be a druggable target. And that subsequently led to a very long journey to look for the right molecule to prevent that glucose toxicity that leads to impaired hypoxia response. And so we started with some key requirements. We obviously wanted this to stabilize HIF-1 alpha. There's also a separate line of evidence that oxidative stress that I haven't presented is also important. So if it could reduce oxidative stress, that would be good. We wanted it to be delivered as a small molecule through intact skin to prevent wound formation. This is a tough area because there's not drugs already in it. So the big pharma companies don't see or don't really understand how to develop drugs. And so we thought if we could find one that was already off patent, that would be good. And then we looked at a whole bunch of different molecules. And at the end of it, we found a molecule called deferoxamine, actually was FDA approved about 60 years ago, and is used to treat hemochromatosis and acute iron poisoning. Its excretion is well understood. The side effects are relatively minor, and it was off patent. So it's essentially a generic and cheap. And again, I won't present all the data, but what we found was that DFO actually prevented reactive oxygen species through Fenton chemistry. So that was number 2 in our need statement. But it also prevented the ability of the enzymes that degrade HIF-1 alpha. The prolyl hydroxylases are all iron dependent. So by blocking or binding iron, you prevent those enzymes from working. And so what you get is essentially upregulation of HIF-1 alpha or stabilization of HIF-1 alpha, some of which goes to the nucleus. And because you have prevented the methylglyoxylation, you're able to bind to P300 and you get upregulation of all those genes. And this is, again, those same 2 mice. And now we've made one diabetic, but treated systemically with deferoxamine. And what you find is you get the similar equivalent amount of tissue survival and recovery. That's different than wound healing. And so we then went with just an eyedropper. And this, unfortunately, this molecule has a very short half-life, and it's also light sensitive. But for proof of concept, we basically dropped every 8 hours deferoxamine onto wounds, and we found that we could accelerate diabetic wound healing in a dose-dependent manner. And that was kind of proof of concept that this seemed to impact the pathophysiology of impaired diabetic wound healing. So as alluded to, we are very interested in small molecules. And again, one of the things that I think sometimes is ill-advised in wound healing is we have this big gnarly hole and we pour stuff into it and we expect it's going to kind of somehow magically change the hole. We've kind of evolved to a concept of treating the donut, not the hole, meaning that you treat the normal skin that surrounds the wound, because that's where the healing, certainly the keratinocytes and many of the fibroblasts are going to come from. So if you can overcome the wound healing diathesis in the donut, not just the hole, that might be a better approach. And to do that, again, we needed to come up with something that could get through the stratum corneum, because intact skin is not as easy to deliver drug to. So the challenges were that this molecule deferoxamine is relatively large. It's at the higher limits of small molecules. I think it's 860 Daltons. And because of that, it's very hydrophilic and it's very difficult to get across the hydrophobic stratum corneum. It also has a short half-life. And so again, a fair amount of work that we did, in concert with our chemical engineering colleagues. We came up with a sustained release drug delivery system for this molecule. I won't go through all the details. It's all in the published literature, but essentially using surfactants to create reverse micelles, you can get this molecule through the skin in a very reproducible way. And this is proof that not in a wound, but in intact skin, where we're looking at how resistant it is to ischemia. You see that if we treat with DFO through the skin, you actually get less of a necrosis in response to pressure. As expected, you get upregulation of the genes that you would expect, increases in blood vessel formation, and you actually get either no wound or a more rapidly healing wound. Interestingly, the wound, when it's healed, when it occurs, has higher breaking strength than untreated wounds. So again, this is important in the context of diabetics, because once they have a wound, they're at high risk of having another wound. And so this is the concept of secondary prevention. And again, this was published several years ago. So as we started thinking about what, this long journey to find something to reverse impaired diabetic wound healing, we started thinking about using DFO. We know where foot ulcers are going to form over the first metatarsal head, over the heel. They're already using duoderms and things like that. If we could deliver the molecule through that, that might be a really interesting, therapeutic for diabetic wound healing. We were chagrined by just the understanding and the roadkill that is in diabetic wound healing. Over the past 20 years, there's not been any successful drug trial or protein peptide trial in the past 20 years. And when you look at, the hit rate of all the different verticals, ophthalmology, allergy, hematology, oncology, the hit rates are daunting. They're, 15%, to get through phase 3. For wound healing, it's been 0%. The last thing that was approved through a drug pathway was PDGF, which is not a small molecule. It's a protein growth factor, and that was 23 years ago. So, I actually work in our in our wound center, and the real challenge is that wound healing trials in diabetics have a real inherent procedural component. You have non-compliant patients. Offloading is difficult to control, and the FDA only recognizes complete closure. Because of this, we decided to go after a different wound healing diathesis, sickle cell ulcers. And so there's lots of really translational business reasons to do that. The most important being you can get approval with 2 small trials if you get orphan drug designation from the FDA. You also get some tax breaks and you can set your pricing. So sickle cell ulcers are an orphan disease, only 14,000 per year. You have to be less than 200,000. And there's really, we took this to third-party payers and got, them to say, if we could decrease the incidence of this by 20%, 40%, what would you be the pricing? And the pricing was surprisingly high. For third-party payers, it was up to $65,000 per wound. Again, surprising, but that's how much wounds cost. And so the nice thing about wounds is there's a lot of technology headroom. And so then, we had to show that it worked, and we went, and this is again published in Wound Repair and Regeneration, showing similar to diabetic wound healing, deferoxamine transdermal actually accelerates wound healing in sickle cell ulcers. And so now we're currently in the midst of a FDA. We have an open IND. We're doing a phase 1/2A trial and enrolling at 5 centers across the United States. So, I think this has kind of been, part of what our lab has been doing, which is trying to bring small molecule therapy to abnormal wound healing. I won't talk about, we have a small molecule also for scarring that we're bringing into the clinic. But I think, for those of you starting your labs, I think small molecule approaches are an attractive way to target abnormal underhealing and overhealing wounds, and DFO can help correct the pathophysiology of diabetes to improve healing. These are all the people that have worked over the past 15 years and the funding sources. And with that, I will close and open it up to any questions people might have.
All right, thank you, Jeff. That's a phenomenal talk, and I think there have been several people around the country, myself included, that have definitely benefited from some of the groundbreaking work that you've been doing with deferoxamine. I encourage anybody with questions to either raise your hand or speak up, and I guess maybe I'll open up with one question first. I think it's very interesting what you noticed with your diabetic wounds. demonstrating some increased mechanical strength. And I know that with HIF-1 alpha, sometimes in other pathophysiologic states like pulmonary fibrosis and liver fibrosis, that HIF-1 alpha may actually be involved in upregulating some of that collagen deposition. Do you think deferoxamine may be usable in your patch with, say, regular wounds that may be under some tension, regular surgical wounds that we're closing? Do you think that may help accelerate or increase the strength of these wounds as they're healing?
Yeah, I mean, it's a good question. And you know, we've thought about, you know, different places to try it. And I think one of Kellen Chen, I think a year ago, started to do some experiments along in that area and had promising results, and then other things were more promising. So he's moved on to other things. But yeah, I think you know, that's a nice thing about finally getting something out into the clinic and having it, you know, go through an IND process and have FDA, you know, approval. That then now that you have this device, you can now use it, or not this device, this drug. You can now use it in other clinical areas because you've shown that it's safe, or at least the trial has been approved by the FDA. And so I think there's lots of potential applications that we've been thinking of, and it's really just a question of bandwidth in terms of what we can pursue.
All right, are there any other questions out there?
Dr. Wan, would you mind if I go ahead and ask a question?
Yes, please.
Dr. Gertner, for the delivery, for your transdermal delivery of all small molecules, does this reverse micelle formation help with all types of small molecules, or is it specific to DFO?
No, it's good for hydrophilic small molecules. So we have, you know, looked at it, and actually the small molecule we're using for scarring, we use the same technology. So it seems like it's a very you know, generalizable way to deliver small molecules through human skin.
Is there a size limit to these reverse micelles? Can they deliver larger proteins?
I don't think so. We haven't looked at that, but I think not. I mean, that's, you know, there's lots of companies that have claimed to be able to do that and then have failed. And so I would be very remiss to say, I think we can do what everyone else hasn't been able to do. But, you know, I think it's, you know, we've looked at it at a very superficial level, and it doesn't seem like it's going to work for proteins and peptides.
Are there any questions out there? Just please raise your hand or just speak up.
All right. I must have wowed them.
All right. Well, thank you. Oh.
Is there time for one more before
Yes.
I talk?
No, please.
Thanks for sharing, Jeff. It was a great talk. When you pivoted from diabetic to sickle cell ulcers, it was strategic, but I was looking up sickle cell ulcers because I don't treat them, so I'm not that familiar. Is there some shared biology that explains why DFO works? And then sort of as a offshoot of that, you know, could DFO work for all sorts of wounds, venous stasis? You know, what's the limitation? Like, what won't DFO help with?
Yeah, I mean, we think it might. And again, I don't want to say this is like a miracle drug that works for everything. It does seem to have salutary effects on many things that are involved in cutaneous biology. You know, for purposes of the FDA, no, it only works for sickle cell ulcers. Otherwise, we won't have orphan drug designation. But you know, the strategy is to kind of get it into the market, get some sort of commercially viable business, and then, you know, you can explore these other areas. And you know, our model is, well, you can make enough money from an orphan indication that then you can fund these other trials. Once you get approval for any other indication that's not orphan, you lose your orphan designation. But, you know, at that point, we would have been able to self-fund trials. So I think it is, I think, you know, there's a lot of evidence that iron in the skin is part of the path, or not a lot. There's intriguing, you know, suggestions that iron in the skin from extravasated blood leads to, venous leg ulcers, or at least, some of what we see. Because I don't think it's as simple as like, oh, it's just, like a hot dog exploding when you heat it up. It's more because the things don't heal. And I think all of us who've ever dealt with venous leg ulcers know, it's really challenging. Even a small one, you do all these things, you put them in an Uniboot, and the damn thing never heals. And so I think there is something about, dermal and, subdermal iron overload that might be a common pathway, but that's not for the FDA to hear.
Great, thanks for sharing.
All right, thank you. All right, so we can move to our second speaker, Dr. Alex Wong from USC. He's an associate professor in plastics and reconstructive surgery, and he serves as the director of research at USC, as well as the program director for the Microsurgery Fellowship. He's been part of the PSRC for nearly 15 years, and he currently serves on the executive committee as parliamentarian. He previously chaired the technology committee, and he currently chairs the ASPS Educational Network Committee on Research and Technology. Alex runs an NIH and PSF-funded research laboratory focused on pathobiology of non-healing irradiated wounds and lymphatic regeneration. And so tonight he will be speaking on his work with wound healing in the irradiated setting. Alex?
Thanks, Derek. Thanks for the opportunity to speak. I'm going to share my screen here. Okay, can you see my screen?
Yeah.
Great. So these are my disclosures that aren't related to the talk. So wounds, very tough, and they're all sort of different. When I started practice in plastic surgery, I saw a lot of radiated wounds just because of the environment I'm in, a lot of sarcoma resection, breast reconstruction, and I found these particularly challenging. And here's an example of someone that walked in literally a couple weeks ago with a very challenging problem, a woman that had breast cancer on the left side and was stable for a while, and then had a spontaneous ulcer literally 10 years after her radiation. So clearly a difficult problem. Another patient that had sarcoma of the leg, wide local excision, and then reconstruction with a vertical rectus flap, and did actually really well for 5 years, and then spontaneously came in with this 2x2 sized ulcer at the middle of the flap, probably where they delivered the radiation. Again, difficult to deal with. And, radiation wounds, they just keep getting worse, and are always a risk for patients. And this is a patient who had sarcoma resection and reconstruction 40 years prior, and then ended up with this very difficult ulcer. So a challenging problem, in my opinion. It's characterized by fibrosis, some skin color changes, dry skin, and you end up with this non-healing ulcerated wound that is difficult to manage. Some people choose to just have these open wounds for months or even years, but when they come to a plastic surgeon, we're given an ice pick or a hammer, and the one we like to use is a scalpel. So generally we like to debride these to the extent of the margin of the radiation to get to some good tissue. But unfortunately, at the base of these wounds, not only is the skin damaged, but the underlying fat and muscle and everything has been damaged. So it is not a good bed for anything, doesn't support a skin graft, and doesn't heal on its own. So, for this patient, really the only option was to bring in some non-vascularized tissue, and we did a free latissimus flap to the posterior to the popliteal fossa. And while it looked pretty straightforward here, believe me, from this to this flap, it was literally probably a year and a half before his wound stabilized. So there was a lot of local wound care in getting this flap to heal to this radiated area. So although this picture looks straightforward, it certainly was not. But radiation is likely to be an increasing problem for wound healing and probably some other medical or surgical conditions due to the increasing cancer incidence. In general, people are living longer, healthier, and as they age, they're at higher risk for cancer. So if you look at data from 2016, there were about 15 million cancers in the US, and By 2026, 10 years after, this is going to be more than 20 million cancers, and about half of the solid tumors that people experience require radiation for its management. So this is going to be something that we're going to be managing more as plastic surgeons. And so what are the goals of radiation therapy? Mainly it's to reduce the recurrence. It kills the cancer cells that surgery can't take care of. But unfortunately, the side effect is that the healthy tissue surrounding the area is definitely damaged by the radiation. And the mechanism of action is by direct injury to the DNA, causing double-stranded DNA breaks, as well as injury to the surrounding water molecules, which launch free radicals and cause further damage to the DNA. Radiation injury can be characterized into a couple different phases: immediate, which occurs hours to days after the injury; an acute phase, which occurs days to weeks after; and then the delayed injury, which occurs months to many years after the injury occurs. And usually it's the delayed phenomenon that requires surgical intervention, because the immediate and the acute are self-limiting. Immediate injury, here's a picture. It looks like transient erythema, as well as some epilation and dry skin. It subsides on its own in a couple of days and can be treated with local moisturizing lotions and protection from the sun. Acute injury happens a little bit later, in the 3rd or 4th week. This is larger doses of radiation, and it appears as, looks a little bit like cellulitis. It's edematous, and there's sometimes some hemorrhage due to the injury. This also generally is self-limiting. In some cases, it can become either dry or moist desquamation, depending on the dose. These can last sometimes up to 2 months. The moist desquamation is painful, characterized by bulla, essentially is a radiation burn. But again, with local wound care, these pathologies can resolve on their own. And here are just some other ways to deal with these. Wearing loose clothing helps. Managing drainage with different dressing is important. And avoiding sun or heat exposure can help. But most of the talk is going to focus on what comes to us as reconstructive surgeons. The acute and radiation injury is managed by nurses and radiation therapists, and usually, as I mentioned, are self-limiting. But these are the chronic wounds that end up stumping everybody else, and they end up in our offices. And these heal poorly due to decreased vascularity, local immunosuppression, and also injury and necrosis of deeper structures such as bone or tendon that become infected and complicate the situation. And so, as I mentioned, delayed effects of radiation appear 6 to 10 weeks after the injury. These are correlated with higher doses of radiation, and patients develop either hyperpigmentation or hypopigmentation, depending on how the melanocytes react to the injury. But there is certainly fibrosis, atrophy of the skin. Sometimes you'll see telangiectasias, and then sometimes you'll see ulceration associated with this. And why do you have atrophy? It's because of decreased fibroblast activity, resorption of collagen. Why do you have fibrosis? It's because of the injured fibroblasts are disabled or not working correctly, and they deposit dense fibrous tissue in response to TGF-beta and other cytokines. Why is there dryness or lack of hair growth? Essentially, the radiation destroys the sebaceous glands and hair follicles. And then why do you have these pigment changes? Again, it really depends on how the melanocytes respond to radiation, and you can have either hyper or hypopigmentation. And this is a summary of the different things that can happen in terms of response to radiation, including cellular depletion, changes in the extracellular matrix, microvessel disease, fibroblast dysfunction, and changes of the cytokine environment. All of these contribute to chronic fibrosis and slow wound healing. So this is an example of more things that would walk into our office as plastic surgeons. This is a woman that had a sarcoma resection and radiation and had this open wound for over 1.5 years. She treated this with conservative therapies, such as weekly debridements and topical and antimicrobial dressings at a wound care center. She also tried hyperbaric oxygen over 120 dives, which is probably well beyond the norm. She tried different therapies. As Dr. Gertner suggested, the only sort of biologic out there is PDGF, platelet-derived growth factor, or Ranix. This failed, and so she came to me. Essentially, in my practice, this requires debridement and reconstruction with tissue that is not radiated. In this case, we chose a free gracilis muscle and skin graft and plugged it in distal to the zone of injury to the anterior tibial system. Another patient I saw that sort of inspired some of the work I'll talk about in the next 10 or 15 minutes, woman with a spinal tumor, prior VY flaps for this spinal tumor. She had adjuvant radiation and then ended up with this ulcer and sacral osteomyelitis. We ended up debriding this and got rid of all the radiation tissue and removed the sacral bone. Unfortunately, when we went back to reconstruct her, she had an intraoperative MI, and we had to vac her for 6 weeks. And she had a CABG during this time. And once she recovered from a cardiac standpoint, we planned a posterior gluteal thigh flap and rotated this. This is based off the descending branch of the inferior gluteal artery, and essentially rotated it into the defect and skin grafted the posterior thigh donor site. And you can see the proximal portion of the flap here and the distal part here. We also re-advanced the right-sided VY flap, and this is the long-term sequela. Certainly she's been through a lot, but she has a staple wound, and that was what's important to her. Another patient with radiation to the knee and sarcoma. She had this patient had a wide resection, and the extent of the radiation was so large that really there was no single flap donor site large enough to cover this latissimus ALT. Very difficult to cover this whole thing. So we didn't remove all the radiation. We left some of it, as you can see out here. And this was a very large anterior lateral thigh flap, but you can see there was still remnants of the radiated tissue. We went back and contoured this and liposucked it down and removed some skin, but during the wound healing process, still at the edges of the vascularized flap that did not have radiation and the flap that and the residual tissue that did have radiation, it was just very difficult to heal at this junction. And so this prompted, some questions in my mind. And basically, as researchers in plastic surgery, we have the opportunity to try and take some of these problems and go to the laboratory and synthesize them and ask questions about the biology with the hope of coming up with some therapeutic targets, and then again, moving those through the clinical research cycle and then actually having real treatments that patients can use. So, the questions I thought about were these smaller wounds, not these large ones that required free flaps. Those are actually relatively straightforward. These small ones, occur in patients that really are not, you wouldn't do a big operation for this. Sometimes they're in elderly patients that have comorbidities that can't have a big free flap. So what do you do with this? And then also it's the junction of the big areas of radiated skin and then the new free flap that you bring in, as I saw in the last case example. There's little parts of the wound that just can't heal. So the questions were, what is dysfunctional at the cellular and molecular level of this radiated skin? And the second one was, besides the molecular changes, are some of the stem cells in the skin also defective? And then basically, if we explore this, can any of these things we learn in the laboratory be used to tailor new therapeutics? So this is what I call the bedside or operating room to bench approach. So here, since I had all these patients with radiated wound problems, I had an opportunity to capture the skin in an IRB approved protocol. And since we were removing the radiated skin and reconstructing the wounds with flaps with normal skin from the same patient, we had paired samples and kept this. We then isolated the protein from the skin, and this was probably 8 or 9 years ago before some of the advances in proteomics. So we used at this point, somewhat archaic technology called reverse phase protein assay that identified differences in proteins between these radiated and normal skin samples. To address the second question about are the stem cells deranged, from the same samples, we isolated the skin mesenchymal stem cells with the help of one of our neighboring labs that had expertise in this. And then we used more traditional techniques to isolate the RNA and then use RNA sequencing strategies to identify differences in transcription patterns of the MSCs to find out more. So in terms of the first part, we did this and we just wanted to confirm that the radiated skin was indeed damaged, and we did. Showed the typical changes in radiated skin, such as thinner epidermis and deranged collagen, as stained by Sirius red. And here was the results of the proteomic analysis. I apologize for some of the blurred out lettering, but we looked at a series of normal skin and radiated skin. And from all these, we found that the most downregulated protein was here in the top. And this is a heat map where if it's blue, that means there's not very much of it. And if it's red, there is a higher level. And the top protein that we found that was downregulated in the radiated skin was fibronectin. And fibronectin is probably well known to most of us from our cell biology and histology days. It's an extracellular matrix protein. And here we did some Western analysis to confirm that indeed fibronectin was downregulated in radiated skin with radiation indicated by the plus sign. And in each patient, it was always lower relative to the non-radiated control. This was also verified by quantitative RT-PCR. So, with that in hand, we wanted to test the hypothesis of, well, if radiation leads to lower levels of fibronectin, a sort of straightforward question would be, if you do this in another experimental model, is that true? Meaning, is fibronectin downregulated? And then the next logical question for our group was, well, if radiation leads to less fibronectin, what would happen if you give more fibronectin back? You would think that maybe that would help. And so we did this, and we utilized a radiation model that was delayed in fashion. So we radiated mice with 20 gray and then allowed them to recover in our vivarium for about a month to simulate more of a chronic model as opposed to an acute injury model, and then perform standard wounds on them. And here, if you compare normal mice that did not receive radiation to those that did, you can see that the radiated mice heal much slower, as indicated by the fact that the percent of wound area always lags that of normal mice. And so here was the basic question, well, if you give radiated mice more fibronectin, does that help the wound heal faster? And it actually does. On the top, you can see control wounds treated with a control vehicle. And then on the bottom, the time-matched control at day 0, 14, and 21. And you can see that at day 14, there is improvement in wound healing. And at day 21, the wound is healed, whereas the non-treated, the control wound still lags and has an opening there. And this was a one-time application of fibronectin. We found that when we looked at the wounds, that it was characterized, or the fibronectin treated wounds were characterized by less neutrophils, less macrophages, and slightly more endothelial cells. So that gave us some insight and one potential molecule to explore further. I'll transition a little bit to question 2 before I get back to the fibronectin story. And the question is, are stem cells also damaged or defective? And since we had all these paired skin samples, we isolated skin MSCs. And again, this protocol is published and it's not anything particularly special. You basically mince the tissue and digest it, and then the MSCs end up as the population that sticks to tissue culture plastic. And then we characterize this these cells and verified that they had the typical immunophenotype for skin mesenchymal stem cells, as shown below here. And when we looked at the skin MSCs from normal skin and compared them to the skin from the radiated skin, there were some pretty gross phenotypic differences. The cells just were larger, they looked more abnormal in general, and there was a lot more variance in their cell shape. And then we tested whether or not skin mesenchymal stem cells from the radiated skin were different from skin that was normal. And not surprisingly, they were. if you push these skin MSCs to either the adipocyte or osteogenic lineage, they have a decreased capacity to differentiate. And then when you perform something called a colony forming unit assay, or CFU assay, essentially you can take about 100,000 skin MSCs, plate them into a tissue culture plastic dish, and then days later stain the colonies with dye. You can see that skin MSCs from normal skin have a pretty robust ability to form colonies, whereas skin MSCs from radiated skin really are not able to do this well and are significantly disabled and therefore non-normal. Finally, we wanted to see if these skin MSCs could migrate normally, and this was a gold salt assay that was performed in conjunction with some of our collaborators here at USC that do this frequently. And essentially, you can imagine this white area as gold salt, and you basically put cells on top, and the black areas are where the cells move, and by migrating, they chew up this gold salt. And so the more black blobs you see essentially correlate with migration activity. So if you compare normal MSCs from skin, MSCs from normal skin, this is the baseline. On the far left, this is fibroblasts, which are the control for this assay. And then if you look at MSCs from radiated skin, you can see that essentially the black area is significantly less, indicating that they're just not very good at migrating within this gold salt. We also did some RNA-seq. Since we're running a little short on time, I'm going to advance through this, but I'm happy to discuss this further. Essentially, we got to a point where we found from our PPA analysis of skin that low fibronectin was associated with radiation damage. We showed that an exogenous single dose of topical fibronectin improved wound healing in a radiation wound model. And essentially, we wanted to push this further towards clinical translation. And then the skin MSC analysis, I could talk more about it another time, but since we're running short, I won't discuss this. So essentially, getting back to the fibronectin story, this was a bedside to bench initiation. And we made some good progress, proved that it was helpful in our experimental models. But really, there's a significant challenge in going back to the bedside or 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 clinic based on this bench data. And so this is a story of how we translate this into humans. And in the previous talk also discussed the challenges of that. And I think it's great that we're focusing on this, because although there's a lot of great basic science that we're doing as plastic surgeons, the more impactful thing is to take some of our findings and push them back and impact patient care. And so this was helped along by something called the Center for Dental, Oral, and Craniofacial Tissue and Organ Regeneration. This is an NIH-funded program that awarded us some money to work on this problem. It also supports Dr. Wan and Dr. Gertner and a bunch of us in the California institutions. So very grateful for this. And I collaborated with Bohan, who is an engineer here at USC, and she has this patented formulation for a hydrogel formulation. And we thought about combining our fibronectin finding with this hydrogel that was able to deliver fibronectin in a slow release format to try and improve the efficacy of it and translate this into a real product that could help people. In particular, we focused on patients with irradiated head and neck wounds to address the needs of C-Doctor in particular, although the findings certainly could be applied to radiated wounds elsewhere. So although we had some interesting findings in rodents, you can do a lot of things in rodents. You can probably cure aging, cure cancer, cure a lot of diseases. The onus is on us as investigators as we translate this into things for humans, is to prove that the biology can be confirmed in other species. In our case, since this was a skin wound healing phenomenon, we've chosen to go to swine. We and others have believed that or know that pig skin is more similar to human than rodents with respect to their epidermal thickness, their lipid composition, their collagen fiber arrangement, Pigs are not loose-skinned and are very different from rodents, which are very loose-skinned, so that affects wound healing biology. And also, rodents are known to heal by wound contraction and not epithelialization, which is very different from humans. Moreover, pigs have a more similar metabolism, and so for both ingested systemic or topical drugs, their metabolism is more similar to humans, so they were a natural choice to go to, and that's what we set out to do. And pretty similar to our mouse model, we scaled this up such that we were now using a larger animal, such as a mini pig, and essentially this was a fractionated dose of radiation, 5.5 gray over time, and this was developed based on prior literature from the Michigan group, as well as some of our radiation oncologists, that ensured that this was close for a clinically applicable radiation wound. We allowed these pigs to recover for 6 weeks, again, to simulate more of a chronic wound injury as opposed to an acute one, and then essentially created wounds, followed them carefully over time, and then harvested them at 1 month after wound creation and treatment for further analysis to essentially ask the question, does fibronectin work in a different species such as pig? And the short answer is that it does. On the top, you can see the control hydrogel treated group of pig wounds. And then the bottom, this is the fibronectin and hydrogel treated group. And you can see that by day 14, you can already see a fairly obvious improvement in the wound with the fibronectin treatment. And this continues to be significant at days 21 and day 28. And this is just a graph that confirms that and shows the statistical differences. So we were interested also in trying to figure out, well, why does fibronectin work? Although we showed that in some of our mouse data, it's well known that radiation increases mediators such as transforming growth factor beta-1 and SMAD3. This is well known, not something discovered by us. But we wanted to confirm that this was happening in our pig radiation models. And we demonstrated that in the radiated tissue, there was an upregulation of TGF-beta-1. This is indicated in panel A as a semi-quantitative analysis of the immunohistochemistry. We then did the same for SMAD3 and showed that, again, radiation increases SMAD3. And then as another way to be more quantitative, we did an ELISA for the protein from the skin. So we took skin from both normal and unirradiated tissue, ground it up, extracted the protein, and subjected this to an ELISA, and found, again, at the protein level, there's more TGF-beta-1 after radiation. So to further characterize this, we looked at the wounds under the microscope and looked at the H&E, stained it for different things such as Sirius red. We found that fibronectin improves the scar. When we looked carefully at the area, while there's disorganized tissue in control radiated wounds, the fibronectin treated groups did show more organization and less scar. And this is confirmed by the Sirius red as well in terms of the more organized collagen fibers. And then going back to TGF-beta-1 and SMAD3, which we know are elevated after radiation, we wanted to see if fibronectin treatment could modulate this. And indeed, if you look at the immunohistochemistry with standard brown indicating a positive signal, you can see that in the control wounds, there's a fair amount of staining for both TGF-beta-1 and SMAD3. And if you look in the fibronectin-treated wounds, there's a significant decrease in both of these mediators. And so, what's the road to making this into a product? It's a long one, as I'm finding out. I've spent the first 8 to 10 years of my laboratory life understanding how to make experiments work in the lab, and we've gotten okay at that. But we're transitioning to really taking some of the fruits from our findings and getting this to helping patients, and certainly entities like SeaDoctor and others are helping us do this. And I think this is an exciting time for us as surgeons, scientists, and investigators. But the road to getting a drug to the FDA is long. I think Dr. Gertner alluded to that. I certainly am learning about this and doing everything I can and reading as much as I can to figure out how to do that. But fortunately, we do have some experienced people in our field that we can look to for leadership. And what are we doing? Basically, we're at a point where we're not just mixing these things up in the lab and putting them in a water bath and then putting them on pigs. I mean, this works in the lab and it's great, but this is not a patient-friendly construct. We have to get this laboratory sequence into something that's reproducible, standard. So we're going to take these components and put them into a double barrel syringe, something that can be sterilized, frozen, and then turned into an off-the-shelf product. Easier to say than to do. We have to identify the appropriate user-friendly apparatus, scale this up, do this in with GMP sourced materials or good manufacturing practice materials, which are much more expensive than the ones you would order from Sigma or another company that you would use for laboratory or animal products. And then do this in a testing facility that can assess biocompatibility and sterilization. All these things need to be done before you can even bring this up with the FDA. This just shows how much work there is in moving from the laboratory and into a translational setting and then getting it to patients. But certainly I think this is a really important goal for us to all keep our sights on. And so hopefully we can update you on our story in a couple of years. So I've talked about radiation wounds, talked about the challenges that are associated with them. There's no doubt that this will increase as cancer increases. So I believe this is something that we should definitely invest resources to. I touched a little bit about the different phases of radiation injury, talked about some surgical approaches to chronic radiation wounds. And as I mentioned, in our hands, this is straightforward. Not that surgery is easy, but we know what to do. We need to remove the tissue and replace it with a flap. But understanding the pathobiology of radiation damaged skin, I think it's a large task. Again, a similar parallel to the diabetic wounds that Dr. Gertner talked about. Very, very complex. We're just skimming the surface of this. And we've focused on one molecule that looks like it's different, but I'm sure there are many, many more that we could interrogate and potentially combine with fibronectin that could make this even better. And then finally closed with clinical translation of this bedside to bench and now back to bedside process showed that topical fibronectin robustly improves healing in our clinically relevant models, seems to improve wound quality, and it decreases some of the inflammatory mediators associated with radiation. So we're excited by this and hopefully you learned a little bit about the process. I'm happy to take any questions. Apologize for going a few minutes over, but again, thank you for the opportunity to share some of our work. And then this is, of course, this is a multi-year process held by many, many individuals from my laboratory over many, many years. So I appreciate all the hard work that was done into this, and also coordination with radiation biologists, molecular biologists, engineers. Certainly reach out to everyone around you that can help you solve the problems you're interested in solving.
All right, thank you, Alex. That's a phenomenal talk. I think many of us on tonight have definitely seen these difficult wounds in radiated sites. And I think for all the trainees out there, I think what you're doing, as well as what Dr. Gertner talked about tonight, are perfect examples of how you integrate problems that you see clinically with translational research to really do cutting edge work with potential for tremendous impact. So thank you for your talk, Alex. It looks like we have a question from Lin Vong from WashU. So Lin, do you want to ask your question? Lynn, are you still on?
Yeah, I'm still on. Hi.
Okay.
Hi, Dr. Wong. Thank you so much for the great talk. So I was just wondering, after the fibronectin hydrogel treatments, do you also see like an improvement in regeneration of dermal appendages, like sweat or sebaceous glands or hair follicles? Because I mean, with that will get us even closer to like as normal skin as possible.
Sure, thanks, Lynn. That's a really good question and good observation. We haven't specifically done a really robust assessment of that. I think that would make a lot of sense in terms of implicating fibronectin as a true regenerative treatment. I'm afraid I don't have a good answer for you. I'm also afraid that I think that it may not, although there's some improvement in the scar quality based on some of the H&E that I've looked at. I haven't seen a robust regeneration of sebaceous glands and follicles. So although it does improve the wound healing a bit and decreases the inflammatory radiated pathobiology, it's a little bit short of true regeneration, but we can hope for more. And potentially, my thought is that maybe combining this with some other therapeutics might push that towards a true regeneration. But it's a really challenging problem. I mean, I'm not sure if there's any great therapeutics that can be truly regenerative and regenerate dermal appendages that, as you've mentioned. I think it's a great goal. I might ask my my other expert panelists, Derek or Jeff, if you think that this is a goal that we could really strive for, if you know something that does it.
Yeah,
I mean,
no,
I think it's
go ahead, Jeff.
Yeah, I was going to say, I think it's a worthwhile goal, and I don't see any reason that, you know, a priori it's impossible. I just, you know, I think it's, you know, something that we should, you know, continue to, you know, try to achieve. Again, it should be pretty straightforward. You know, I mean, to you know, skin is a relatively thin organ. If you have the right components, there's a lot of self-assembly that goes on in skin. So I would think that, you know, it should be achievable. I mean, I think it's more achievable than regenerating your brain, for instance. so I think, you know, and I think it's something we aspire to.
All right, great. Are there any other questions? I guess I can ask a couple questions, Alex. You know, again, phenomenal work. It's very much related to some of the stuff we're doing in my own lab here. But I have two questions. Have you looked at identifying like a cell surface marker profile for these skin MSCs, and so that would allow for prospective isolation from, you know, fresh samples for some interesting analysis? And the second question is, you know, with the fibronectin downregulation in these irradiated wounds, you know, it's involved in cell adhesion and migration. And do you think that has changed the mechanical kind of microenvironment niche for these MSCs, impacting their function? Or do you think it's more of an intrinsic dysfunction in these MSCs due to the radiation, like you've shown with your in vitro data?
Yeah, Derek, thanks for the questions. To address the first one, the cell surface markers we utilized are mostly based on literature and the standard MSC markers, at least in my CD90 STRO1, we used to confirm. I think the question, maybe the question was, are the skin MSCs, do they have a more specialized phenotype?
Yeah, or are they more similar just to other MSCs in fat and bone marrow and muscle?
Right. Yeah. Based on the generic markers we selected for, they're pretty similar to what you'd find in fat or bone marrow. And then to get to the other question, you know, we've currently looked at it as sort of separate phenomenons and isolated the fact that yes, the skin mesenchymal stem cells are abnormal, and then fibronectin is dysregulated or down. You know, sort of putting them together makes good sense. I'm sure there's some linkage. The only issue with radiation is it's like a big bomb. I mean, I wish I could say that these were the only things that were different in radiation, and you know this, and I think a lot of people on this call know this. Unfortunately, probably everything in that milieu is set back, whether it's the remaining endothelial cells, the keratinocytes. They're all a little bit slow. We've just pointed out some differences. You know, my hope is that we can identify the sort of critical differences and the ones that are targetable or druggable or fixable, and find the most simplistic way to help these wounds along. Obviously, we could throw everything at these wounds, and they may all sort of help in some way. But I think to develop a strategic, cost-effective, almost personalized approach to different wounds I think we certainly are getting the information and data to do that, and taking each of these wounds and characterizing the up to the minute biology. Were they radiated? Were they venous stasis diabetic? As we understand wounds better, hopefully we can create, we can accumulate this big menu of wound healing factors and then come up with the right formulation for them, almost akin to cancer, this combination cancer therapy that certainly has had a big impact. So I think it's great that we're all contributing in a way to understanding all these different wounds. And then one day it'd be great to have this menu that we can plug in.
Yeah, thank you. Are there any other questions? Okay, that looks like it's about it. And I guess it's about time anyways. So thank you, Alex. These have both been incredibly enlightening talks that we've had tonight from both Dr. Gertner and from Dr. Wong. So thank you both for graciously volunteering your time this evening to speak. Once again, I would also like to thank 3M for their generous support of tonight's program. Now, looking forward, we have part 3 of our webinar series scheduled for this coming Tuesday, entitled Nuts and Bolts, Mechanics of Wound Healing. And we're having both Mike Longacre from Stanford and Ernie Chu from NYU joining us to give expert talks on mechanotransduction and wound healing and clinical studies on biologic agents for chronic wounds. So thank you everyone again for the wonderful evening, and I look forward to seeing you all this coming Tuesday. All right, thank you.
Thanks, Eric.
Thank you.